From Microbe TV, this is Immune, episode number 95, recorded on July 22nd, 2025. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back. How are you doing? You're enjoying your summer?
It all mushes into 1 year. But yeah, Brianna and I just returned from ASV. That was a lot of fun. Also joining us from Madison, New Jersey, Brian Barker.
Hi, great to be here. It's great to be back from sort of a little conference palooza where I was going to a couple conferences back to back.
So palooza is the word when you do a bunch of stuff together?
Yeah, sure.
Yeah, I think that's a good one.
So now it's, I get to think even about more science.
Also joining us from Cleveland, Ohio, Steph Langle.
Hi everyone. Great to be here. Yeah, summer's chugging along nicely. Weather's good. Lots of fun things to do. So good to be here with you all, though.
Yeah, it's the summer of immunology. That's what it's like here on Immune. If you like these programs, by the way, we'd love your support. We don't do advertising to make revenue. We depend on listeners like you. And so your support is greatly needed. We give you science, real science, no lies, no misinformation, no disinformation. There's a difference, right?
There is, yep.
Misinformation is wrong stuff, and it could be unintentional, right?
Yeah.
If I say all DNA is Z-DNA, I would be giving you misinformation. Should know better. But disinformation was you purposely mislead people, like That's saying right. The polio vaccine kills more people than it helps, which is complete disinformation. I mean,
right. Agreed.
You could not make misinformation out of that, because all you have to do is read some papers. Anyway, we give you the science, and for that, we love your support so we can continue. Go to microbe.tv/contribute. All right, today we have some snippets and a paper, and Cindy's going to start us out.
So I was blown away by something I saw in a recent issue of Nature. And basically, the paper was saying that mitochondria, so mitochondria are the powerhouse, basically the power plant of the cell. And they have some of their own DNA, and they encode a lot of the proteins that are involved in generating the power for the cell. And what the paper said was that these mitochondria can be shuttled back and forth between cancer cells and nerve cells. And I just thought, what? I had to do a double take, and I looked at this paper, and then I started digging a little deeper, and it turns out that cancer cells can also steal mitochondria from immune cells. And then I found also another paper that the cancer cells can donate mitochondria to immune cells and modulate their function. And so I just thought this whole idea of stealing and sharing and donating mutant mitochondria was so cool. And I had never heard of this happening, because when we think about it, a cell is encased, and, cells bleb off things, and they can share things. If there's intimate contact between two cells, one can yank off some membrane or some things from the other cell. But I'd never heard about this idea that one cell could basically spit off a straw and suck or spit out individual organelles from one cell to another cell.
They're big.
They're big.
I guess these tubules you're talking about, can suck up something so large as a mitochondria.
Yeah, I guess if you think of was it bubble tea with those little balls in it? Yeah. Basically, it's like that, right? So anyway, I just thought this was the coolest thing, and I thought I would summarize just a little bit of each one of these papers, since I thought it was cool. So the first one is from Nature in 2025, just this past month, and it's called Nerve to Cancer Transfer of Mitochondria During Cancer Metastasis. So this is nerves that innervate cancers, And basically the nerves donate, or the cancer cells steal, however you want to look at it, is cancer cells are stealing neurons or mitochondria from neurons. Now, the first thing you might ask it, well, first is, what? Does that happen? Yes, it does. The second thing is how and why. So the why is really interesting. Cancer growth and metastasis, it's a high energy thing. Like, these cells are growing like mad. They need a lot of energy. And so basically they're like, well, I don't have enough mitochondria to do what I want to do, so I'm going to steal them from another cell. Mm-hmm. They can steal them from nearby cells. It's been shown that neurons can support cancer initiation and growth, but it really wasn't known how this could happen. It was also known, which I just said, is that cancer cells have this really high metabolic rate. So it's a high demand for high levels of mitochondria. They can duplicate their mitochondria on various different things, but exactly how this happened was not known. And basically, it turns out that, cancer cells can siphon off these mitochondria from the neurons, and they can steal them from lots of different cells. But basically, this paper went into a lot of detail and mechanism that I'm not going to go into, but if you want to check it out, you can. But bottom line is, these nerves are sitting right and innervating into the cancer cells, and the cancer cells make these tunneling nanotubules that they go over and pop the straw into the neuron and just like suck off the mitochondria so that they have more mitochondria.
This innervation, is this... So as this happens in a tumor, does it have to be so many cancer cells? Because I'm sure not just one cancer cell is going to be innervated, right?
That I don't know, but I would assume that I didn't go and look a lot into the literature, how much is known about this. But I mean, cancer cells can manipulate their environment. They change the extracellular matrix. They can change the vasculature. So I'm guessing they can also, call in nerves to move over the processes to be able to do this. But there are also like nerve endings all over the body. So depending on where the cancer is, it might just take advantage of that and grow around it.
Yeah. I'm sort of, as you're describing this and as I'm picturing it, I'm really starting to wonder how the tube is specific for mitochondria. Or is it?
Maybe not.
Are other things sucked through the straw? Or does the straw have some way of knowing what is the mitochondria and particularly grabbing it specifically? I'm baffled by this.
I don't know if that level of detail is known. I would assume if you're sucking a mitochondria through a straw, there's probably other things that are going to come with it.
Yeah.
Right. But could you imagine, might there be a time where something other than mitochondria is taken?
I would
what
think
else
so,
do you think there would be? Like, what else? You want the nucleus? You want to take it all?
Well, it's funny you say
know.
that, because I think I've seen information about these kind of tunneling nanotubules being involved in netosis too. So I guess there is an ability of at least some nuclear content to come through one of these little.
I assume it's a pretty wide range. I mean, you could think about like signaling proteins, plasma membrane proteins, microRNA.
Yep. Lots of things I'm sure could move. Yeah.
Fascinating. They specifically looked at this in the brain, correct?
Yeah, they were looking at glioblastoma, I believe.
Mm-hmm. Okay. Yeah. And so it makes sense that the neurons there, it'd be interesting to think about another site where, there are probably not as many neurons, right, to be innervating. But it makes sense for the cancer to evolve this way, because it is highly metabolic itself, to take these mitochondria. I guess I'm surprised that mitochondria are that movable. I thought maybe they would have evolved ways to kind of like, we need to stay here. We're pretty important to the cell. Right.
So that kind of feeds into the next little paper, which was the idea that these nanotubes can traffic mitochondria between cancer cells and immune cells. So I went down this rabbit hole because I was like, what does this have it? Because that first paper really is just about cancer and nerve. And I said, well, that's not really immune. It's not immunology. But what if cancer cells do this with immune cells? And so I went down this rabbit hole, and yes, it does happen. So the the thing with the second paper, so this was in 2022 in Nature Nanotechnology. I didn't give this to you guys, but I'll just summarize it really quickly. Basically, it's called Intercellular Nanotubes Mediated Mitochondrial Trafficking Between Cancer and Immune Cells. And the idea of this paper is, again, that the mitochondrial biomass, as you increase it, enhances functional activities, including T cell functions and so forth. And what they wanted to know was, could cancer cells take mitochondria from T cells, and when they capture them, basically take away the ability of the T cell to use lots of mitochondria? Because not only is cancer cell growth highly energetic, requiring T cell growth and function is also highly energetic, and T cells will switch to a glycolytic generation of ATP because it does it really fast. It's not very efficient, but it will induce a lot of ATP. And so T cells will do this, and they will switch to glycolysis to help with their functions. And basically what this the second paper is showing is that cancer cells can counteract the ability of these T cells to make more mitochondrial mass by basically hijacking the mitochondria from the T cells for their own benefit.
Yeah,
so
and
it's
there's
the
sort
same
of
kind
like
of idea.
It's like it's not just that they are trying to improve themselves, it's that they are trying to actively harm the other cell.
Right, exactly.
Well, cancer cells break all the rules, right?
They do.
So there's no regulation. So it suggests that other cells might do this also normally for different purposes, right?
Yeah. So who knows? I don't know how. I don't know how much it's been looked at, but the paper that I wanted to talk about at least a little bit more is kind of along that lines, in that the cancer cells is taking away mitochondria from the T cells, but then also giving it back mitochondria. So it's this shifting. But the cool thing about this third story is that the cancer cells' mitochondria are actually not working very well because they have mutations. So that's impairing the cancer cells' ability to grow. So they want more mitochondria. They're going to grab the mitochondria that are working from the T cell, but they also then donate back their defective mitochondria to the T cell, and then the T cell holds on to them and now becomes kind of like exhausted and senescent because it loses its ability to do its own energy generation.
Amazing.
Which I just thought was like, what? So
Sort of mind-blowing, yeah.
So this third paper is from 2023 in Nature, and this one is called Immune Evasion Through Mitochondrial Transfer in the Tumor Microenvironment. And this one I just want to spend a teeny little bit more time on. And this is basically is like, we know that cancer cells can create an immunosuppressive environment. And there's lots of things that have been described about immunosuppressive environments. But this idea that T cells switch to this glycolysis, and then they use up all this energy and they become more exhausted. This idea that metabolism and regulating T cell metabolism within the tumor microenvironment might be really important. And the other piece for this story is that the cancer cells have these mutations in their own mitochondrial DNA. And so their own mitochondrial DNA encodes parts of the electron transport chain and other things that are necessary to generate the ATP. And when the cancer cells have these mutations, they don't want those mutations. Their mitochondria are not working well. So they want to steal the mitochondria from their neighboring cells, but then they also want to try and get rid of their own mitochondria. And you might think, well, okay, the normal way mitochondria are recycled or gotten rid of is something called mitophagy. So when a mitochondria goes rogue or it gets mutant or is ready to end its life, basically the cell encapsulates that mitochondria and degrades it down into the building blocks and then reuses those building blocks. So it recycles them. But there's a problem that the cancer mitochondria, these ones with these mutations, don't actually get turned over like that very easily. So the cancer cells kind of get filled up with these mitochondria that aren't working very well. And they want to just get rid of them. They want to give them to T cells. So it turns out, I won't go into too much detail about this, but they did a lot of really cool things with this paper, that they took actual patient tumors, they took out the tumor infiltrating lymphocytes, and they could see that those tumor infiltrating lymphocytes had mutations in their mitochondrial DNA that were the same as the ones in the cancer cell. And then they were able to say, okay, so how do they get those mutations? And they got them because they have this transfer, and they were able to show this in a bunch of different ways. They could label the mitochondria in the cancer cell, and they could see the T cells take up those mitochondria. And they took up the mitochondria whether they were mutant or not. But if they have these mutant ones, they could show they're defective. They could show they don't generate ATP as well, and they could show they could impair the T cell function. And they could show that these were transferred by these tunneling nanotubules. They can also be transferred by extracellular vesicles. So you can just bud off your mitochondria and give them to someone else in the area, which is kind of also interesting.
Yeah, where else are they going? That was the other thing. The nanotubules are not specific to the T cells. That's just what they looked at.
Yeah, so that the same thing happened with the neurons in that first story, so the cancer cells could put out these nanotubes and then suck things through them. I don't know how do you suck things? Like I don't.
Yeah, what are the
how do you
physical
create
forces?
energy?
Right,
The pressure. I don't know how all that works.
And I think because I had been thinking a little bit about some of the things in the paper that I'm going to talk about, I was thinking about, is there like a chemoattractant? But you can't really use a chemoattractant.
Inside the cytoplasm?
Right.
Yeah.
Right, exactly.
Well, I mean, there are because you move proteins around in different compartments of the
Maybe it's
cell.
pressure-based too.
It could be pressure, yeah.
It was so interesting to think about how exactly this was kind of specific, or was it?
Yeah, or was it? But it's a dual benefit to the cancer cell.
Yep.
Because T cells, we're always trying to boost cytotoxic T cells because they're defective and they are good at killing cancer cells.
Yep.
So what a good way, get yourself some more good mitochondria and give your foe something you don't want around, the bad ones.
Yeah.
Fascinating.
And I thought one of the more interesting parts of this was the idea that when those defective mitochondria go into the T cell, they are resistant to this recycling, this mitophagy.
Yeah.
So I started to think, well, is that some sort of vulnerability within the cancer cell that you could potentially take advantage of? I'm not sure how that would work, but it could potentially be something. Because mechanistically, I'm not going to go into the details of this, but mechanistically what they showed was the cancer mitochondria had a protein associated with them called USP30, and that protected them from degradation. And so you can imagine the cancer cell wants to keep as many mitochondria around as possible, unless they're defective. So they are going to limit the amount of mitophagy so they can keep their biomass up of mitochondria and they can grow more quickly. But when they transfer those mitochondria to the T cells, those mitochondria are still resistant to that mitophagy. So the T cell is doing is it's going through its normal process and it's turning over mitochondria on a regular basis. But if you have now these mitochondria that can't be turned over, as you turn over your own mitochondria, they all go away. And what you end up with, what they called homoplasmy, where all these T cells have all defective mitochondria. And that basically makes them exhausted and senescent and poorly functional. So it's just a very interesting way to think about potential vulnerabilities in the cancer cell if you could do something about interfering with the mitophagy so that they did turn over the mitochondria more effectively. So could you target, for example, this USP30 protein so that the cancer cells can't keep their good mitochondria at a high level and it might dampen their growth?
So this, they point out in this paper, this article, that tumors are typically hypoxic, right?
Yep.
So there's not a lot of oxphos going on. It's mostly glycolysis, right?
Yes.
So, I mean, that's not a defect in the mitochondria, although they say later the mitochondria are defective and can't do oxphos, but I thought it was because there's not a lot of oxygen around.
So I think it's two things. There's definitely this hypoxic environment, so cells have to rely more on glycolysis. And I'm not an aficionado in which genes are encoded within the mitochondria DNA and not. But I think that if you have mutations in the mitochondrial DNA, that can actually make some of those proteins that you're now required to rely on in the glycolytic cascade not function as well. So now if you're dependent on glycolysis, but you have mutations in proteins that are part of that machinery that generates the ATP, now you can't generate ATP as efficiently from those mitochondria.
Okay.
Yeah. So I think the $1 million question on this is, can you do anything about this now that we have this knowledge of this sharing these mitochondria? And they do a few studies where they show that there are some drugs that can block extracellular vesicle release or block nanotube formation. And when they use those also with checkpoint therapy, they can have better therapy. So the idea would be that you're restoring the T cell anti-cancer function because you're preventing the transfer of these worse mitochondria to the T cells. So you're allowing them to maintain their energy capacity and their functionality, and they're more anti-tumor.
So using these checkpoint inhibitors, you're doing that?
So you use the checkpoint inhibitors plus something that blocks the mitochondrial transfer.
Yeah.
So you block the mitochondrial transfer, and you also take the brakes off the T cells to allow them to attack the cancer. And there's the little tiny piece of data that they put at the end that basically says that they could find patients, they could do the sequencing of the mitochondrial, or at least search the databases and find out that they could segregate the patient tumors that had mitochondrial mutations from those that didn't. And if you look at the progression-free survival and the overall survival, they survived better if they didn't have these mitochondrial mutations. So I think it's a bit weak, but if you make the leap is that, but people who have cancers, who have these mitochondrial mutations, they can then shuttle their mitochondria to the T cells. And they did show it can happen in vivo. The T cells can take up the mitochondria. And then these T cells are less effective and not able to kill the cancer.
Yeah, it's interesting, the difference between those individuals. It's obviously not everybody has the mutations in their mitochondria, and why is interesting.
Yeah.
But you're right. And then the other question, if you're blocking mitochondrial transfer, would be good to see what other cells are doing that, just so you're not accidentally blocking a cell from getting rid of bad mitochondria that does just normal functions. Like that's a normal function.
Yeah. I want to know more about sort of the normal day-to-day function.
Where are my mitochondria
this.
Going all day?
Yeah.
What are you doing?
Where's my mitochondria and other stuff?
And other stuff. Like shuttling, nanotubules. Yeah.
People must have looked at that in normal cells, right?
Probably. I'm not deep in this specific field, but I would assume that's of interest.
Let's look it up. Horizontal mitochondrial transfer, HMT, occurs between cells. Can happen normally and under pathological conditions. Wow.
But why would cells normally need to do that? That's the question.
Well, I think the cell-to-cell connections, other non-mitochondrial reasons we have talked about before, I think taking each other's receptors to help communication, but specifically the mitochondrial question is interesting.
Yeah. Would that be because you want to transfer signaling molecules, or would that be because you really need to do more metabolism and you don't have enough time to make your own new mitochondria?
So
Maybe you're actually trying to suck other things through and you accidentally pop a mitochondria through the straw.
Yeah.
The bubble tea. Yeah.
I mean, mitochondria do go bad, right? So they
do
be a mechanism to replace them.
But see, that's the interesting thing about it. There must be a specific way to make the mitochondria leave, right? Because otherwise you're losing a bunch of other stuff you may need. There must be some selectivity to the shuttling, or else you're losing your good stuff too, you know?
Yeah, that's the part that I'm so curious about.
Yeah.
So the ways that they are transferred, as Cindy said, tunneling nanotubes, extracellular vesicles, gap junction channels, and cell fusion.
Cell fusion makes sense.
Yeah.
Sure.
Yeah.
Gap junctions, I didn't realize they were that big.
Apparently so.
Mm-hmm.
Who knew?
So
Oh my
cool.
gosh.
Yeah.
But this has been known for 20 years.
Yeah. I guess
But
if you're a cell biologist listening to Immune, you're like, what do these crazy immunologists not know about these basic cell biological processes?
This is what I was thinking. Listen, we don't know the origins of mitochondrial transport. It may be a preserved relic of their alpha proteobacterial ancestry.
Oh,
Well,
wow.
that's the thing.
Interesting.
Which we didn't talk about in the beginning. Mitochondria arrived as they were bacteria that were engulfed into another sac 1.5 billion years ago. So, right, this could be a relic function of that bacterium.
That cancer cells seem to have figured out how to use to their advantage.
Must be beneficial to fitness somehow, and they just came upon it, right?
Maybe they were trying to suck something else out of the cell. They got a mitochondria like, hey, this is pretty good. I have more energy now.
Doing better.
It's evolution, right?
Yeah, exactly.
Yeah.
You walk down a dark alley and get in trouble. That's very cool.
It's awesome.
And that's the beauty of science, is that you find something you like and then you start going down this rabbit hole and look at more and more and more. And that's so much fun, right?
Oh, I had such a blast reading about this because I was like, oh, cancer cells, nerve cells, very interesting. I'm like, does that happen with immune cells? And then I found all this stuff. So,
and then the beauty is on a podcast, you can share it, which is great.
Yes. And it's open science, right?
Mm-hmm. Yeah.
Yep.
It's funny you said 20 years ago, but I'm like, I don't know, 2005 doesn't seem that long ago. It's still in the 2000s.
It's not really that long ago.
I know.
But what's long ago? 100 years?
I don't know.
I don't
it's
know.
all relative, right?
Yeah.
I think the 1950s are long ago.
Yeah, that's a long time.
I could agree with that.
All right. Thanks, Cindy.
Yeah.
Now we're going to talk about kind of similar things from Brianne.
Yeah. So I picked a paper. It's from Science Advances, and it was published earlier this year. This was something that sort of came up in some conversations I've had recently, and I really like this paper because it puts together some sort of clinical observations and some in vivo and some biochemistry, and kind of really goes across the spectrum of a lot of parts of biomedical information. Full disclosure, the first author, Uma Kentheti, is a former member of my lab, and who is now working on this as an MD PhD student at University of Colorado. And all of these authors are at the University of Colorado. But this paper is called PD-L1 CD80 interactions are required for intracellular signaling necessary for dendritic cell migration. There are a couple of sort of areas of background information I should mention before we sort of go into this. One of them, segueing from Cindy's paper, is a little bit about checkpoint inhibitors. And so this is the use of antibodies against some cell surface proteins to block some signaling, usually thought of as blocking negative signaling in T cells. This won the Nobel Prize in 2018. And one of the major proteins that we use an antibody against is PD-1. So anti-PD-1 antibodies are used in a lot of cancer therapies. PD-1's normal function, particularly in the textbooks, is that PD-1 on a T cell will interact with its ligand, PD-L1, on an antigen-presenting cell, like a dendritic cell. And it has been observed that if you use an antibody against PD-1, that's quite effective in cancer treatments. But if you use an antibody against PD-L1, which should just be blocking the other side of this lock and key interaction, that antibody is actually not particularly effective. And so that was actually one of these sort of weird observations that was involved in the backstory of this paper.
Brianne, can you explain why that antibody even works?
Sure. So we think that what happens is that PD-1 when it interacts with its receptor, interacts with ligand PD-L1. I'll say ligand with the L to help everybody out there. That turns off the brakes on T cells. It stops the T cell from being exhausted. And so the idea is that the PD-1 interaction, the PD-1 on the T cell is getting ligated by PD-L1, and that's sort of giving a negative inhibitory signal to the T cell. The cancer cell is wanting that to involve T cell exhaustion. And so if we block that interaction, the T cell might not be exhausted anymore, and the T cell might be better able to fight the cancer cell.
Oh, so the L1 ligand is on the cancer cell.
So the ligand is on the cancer cell, or whatever antigen presenting cell we're seeing here.
It's an evasive maneuver, basically.
It's an evasive maneuver.
But normally we're going to find out where would the ligand be normally?
So the ligand is on a dendritic cell.
And is it function to calm down the T cells after everything is done, basically?
The classic answer is that yes, it's it's calming down the T cells to prevent excess activation.
And that happens also with the CTLA4. So when you activate a T cell, part of the activation process is to start to upregulate the things that will shut down the process and dampen the T cell response and actually clear the T cells later as well. So it all kind of starts at the same time. And this is one of those. So
isn't it a similar function by FAS and FAS ligand?
Kinda.
Because I know that the aqueous of the eye and the brain have a lot of FAS ligand to prevent T cells from screwing it up, right?
Yeah. The signaling details are a bit different. PD-1 and PD-L1 are co-stimulatory molecules, so they influence the TCR signaling a bit more directly.
Yeah.
But the concept that you're saying is entirely correct.
All right.
Yeah. FAS engagement by FAS ligand will often cause the cells to die. So that's one mechanism we call in T cell biology activation-induced cell death. So as a T cell is activated, it upregulates FAS, and then when it engages FAS ligand, then that T cell gets a signal to die. Interestingly, the T cell also upregulates FAS ligand, and that can communicate from cell to cell. We call it fratricide.
Mm-hmm.
So T cells can kill other T cells, but kind of interesting related to what you're going to talk about with this paper is the same cell can upregulate FAS and FAS ligand, and I can signal on the same, I'm moving my fingers together because on the same cell it can engage, which is kind of rather interesting.
The PD-PDL1 does not end up in apoptosis. It just
it's more of a break.
Well, it's more of a break. I mean, PD was originally described as programmed death.
Yeah.
So it can be involved in some apoptosis, but we usually think of it as more of a break.
Okay.
And Brian had mentioned that, blocking PD-1, it doesn't work for all cancers. So there's a lot of other factors. I mean, the levels of PD-1, immunosuppressive cytokines, inhibitory pathways, but then there are just a lack of antigen presenting cells or T cells that infiltrate that are required. So it's, it does work for some cancers, but it still leaves there's so much room, which is where a lot of the cancer field is trying to understand how do you bring those cells in? How do you decrease the immunosuppressive environment?
Yeah, absolutely. But I think that the general idea of why does it not work if you block the PDL1 side, while it does work if you block the PD1 side, is a weird observation.
Mm-hmm.
So Vincent sort of...
I just want to ask one. So Cindy, you mentioned fratricide, but do you know when a sister kills a sister, what that's called?
No, I don't.
Isn't that sororicide?
Yeah.
Oh, it is? Nice. That's a Jeopardy word.
I think we should use them equally.
Yeah. Yes. It's true.
They assume T cells are men or boys.
We say sister daughter cells. We give a gender. We say sister cells, daughter cells. So it should be what is it?
Sororicide.
Sororicide. Interesting.
Wow. Sorority. That's a Jeopardy word?
I don't know. I was just joking. It's a big word that I didn't know. So,
and that Brian did. And she's a Jeopardy winner.
So Vincent brought up kind of what happens in sort of a normal situation with these molecules, and they're all involved in dendritic cell activation. We also need to remember that dendritic cells are found a lot in tissues in a sort of non-active or naive form. This paper, they're going to be thinking a lot about the skin as the tissue. And those dendritic cells can be activated and migrate to a draining lymph node, where they're going to carry antigen that they've uptaken, and they're going to present antigen to T cells. And so that process of dendritic cell activation and migration to the lymph node is key here. Oftentimes, when that dendritic cell gets activated, it starts making co-stimulatory molecules like CD80, also known as B7-1, CD86, known as B7-2, potentially PDL1. And so all of this process of the dendritic cell moving to the draining lymph node, which is key for activating T cells and turning on these co-stimulatory molecules, is also going to be key to this paper. And so one of the things that they talk about here is that, as I mentioned, there's a difference between the anti-PD-1 antibody and the anti-PDL1 antibody. And they have some previous data that there's something going on with these molecules and migration in mice. So the first thing that they do is actually look at some human cells in a transwell. So they're basically looking at the ability of human dendritic cells to move from one chamber of a well of a plate to another.
Through little holes?
After stimulation. Yeah, little holes. So they stimulate the cells to activate those dendritic cells and look at the ability of the dendritic cells to move. If they treat the dendritic cells with an antibody against PD-1, and it's the same one that's used in cancer patients, they see no change in how the dendritic cells move. But if they use the antibody against PDL1, now the dendritic cells aren't moving. And so it seems as though blocking the PDL1 side of this interaction impairs dendritic cell movement, while blocking PD-1 does not. And they further go ahead and look at some biochemistry here, because there are actually 3 different anti-PDL1 antibodies that have been approved potentially for use. And they say, well, perhaps this is related to the affinity of the antibody for PDL1. And they do some experiments where they use these 3 different anti-PDL1 antibodies at different concentrations, and they actually don't see what they expected. They find that the weaker binding antibody is the one that's best at interrupting migration. And they go ahead and actually look at the epitopes and find that the epitope of this antibody that particularly blocks migration blocks a site on the PDL1 molecule that was previously associated with PDL1 interacting with CD80, the B7 molecule, on the same dendritic cell. So we usually think of these molecules interacting with a partner on another cell, which they talk about as being a trans interaction. And we think about, PDL1's job is to interact with PD1. But here they see that this migration phenotype is really blocked when you have an antibody that blocks PDL1 on the dendritic cell from interacting with CD80 on the dendritic cell.
On the same dendritic cell.
On the same dendritic cell, as they talk about it in cis.
Right.
And so the fact that these co-stimulatory molecules have to be on the same cell interacting with one another and are not just interacting with the partners on the other cell was something I hadn't really thought about before looking at this paper.
And that's the terminology cis and trans that Brianne brought up, that you can have, it's the same cell or it's another cell, the interactions.
Yeah. And so I always thought about all of these molecules, B7, PD1, PDL1, CD28, as being molecules that did all of their work in trans. And this is showing potentially that there's a role in cis as well.
But if you think about it, especially a dendritic cell. So it's called a dendritic cell because it has all these dendrites, right? And especially if you take it out of a tissue and you put it in culture, it's going to be floating around with lots of long dendrites attaching down and things. There's nothing necessarily prohibiting it from folding back over on itself.
Mhm.
Sure.
So you really, how would you distinguish a strong side neighbor binding being cis or a folding back over, and
So
it's
they actually
sort
do
of
some of that.
right? It's actually sort of trans because it's the same cell, but it's from far away. I don't know what kind of analogy I could make to that.
Yeah.
It's like if
it's basically a question of, do the actual intracellular parts, do the inside parts of the protein need to be next to each other?
Right.
Or is it just about the extracellular parts being in proximity to one another?
Because if it were just the extracellular parts, you can imagine that you can engage a ligand on a nearby cell, or you could engage the ligand on yourself just because your membrane wraps around and has enough flexibility. And those are different from a direct interaction between the two molecules where you're going to bring the signaling tails close in proximity.
Fortunately, they think about these things.
And have tools to address it.
Yeah, they've got some really cool tools here.
Yeah.
So now they move into a mouse model so they can look at some in vivo experiments. And in this first set of experiments, they paint the dye FITC onto the skin of a mouse and also stimulate with poly-IC to activate dendritic cells. And then they look to see how many dendritic cells have moved to the lymph node that have picked up the FITC. So they're basically looking at, they're painting the skin and looking at how many dendritic cells with paint on them make it into the lymph node.
Now, I wondered about this experiment because I haven't done these experiments, but if you paint something on the skin, in theory, it could also, if it's taken up, drain to lymph and get to the lymph node that way. So I don't think they did any experiments to kind of look at that, but I guess there's enough in the literature already establishing this model that primarily you're looking at dendritic cells that are picking it up in the tissue and taking it to the lymph node rather than just draining in the lymph.
They also switched to another model after this, partially because this model induces quite a lot of hypersensitivity in the mice. Yeah. But I think that the other model also starts to address that a little bit.
Because that one's light, right? So that when you shine light, and when you shine light, those dendritic cells that are in the tissue right then will change colors.
Yep.
And so then you can distinguish cells that are resident in whatever tissue you're looking at versus ones that got photo switched and moved to the lymph node.
Exactly.
So I think the combination of two experiments provides enough data to believe that this process is actually happening.
Yeah, exactly. So they first do the skin painting experiments, and they use an antibody against PD-L1, and they show that they get less dendritic cell migration into the lymph node if they block PD-L1, indicating that the PD-L1 is involved in dendritic cell migration. They also do the same experiment in B7 deficient mice who are deficient in both CD80 and CD86, and they see the same defects. And so if their idea is that it's this cis interaction between B7 and PD-L1, the first piece of that saying that they're both involved is supported here, or is at least starting to be supported here. And then they switch to the other model, as Cindy mentioned, where they actually are using light in these mice. So these mice they're called the KIK-GR mice, and they're photosensitive dendritic cells. And so the dendritic cells, when they're hit with violet light, will undergo a change and will now be red. And so you can see which dendritic cells were previously in the skin that have moved to the lymph node. So they do, again, the same experiment in this model, and they show that if they block PD-L1, they do not get dendritic cell migration to the lymph node. and if they use a version of a mouse with PD-L1 with a mutation that has, PD-L1 has amino acid changes as a result of a mutation.
Very good.
Thank you. So that the PD-L1 residues that are thought to be important for interacting with CD80 are mutated, so they can't get this potential cis interaction. Those mice also don't have dendritic cell migration. Again, further saying, okay, we need PD-L1, we need CD80, and in particular, we need these amino acids in PD-L1 to get dendritic cell migration.
So these antibodies are used for therapeutics of certain cancers, right? Mm-hmm. But wouldn't inhibition of migration be bad for that?
Well, so that's the idea, is that before this, we didn't really think of these proteins as being involved in migration. Right. And these antibodies don't seem to work for cancer, while antibodies against PD-1 do work. Mm. And so perhaps the reason why these antibodies aren't working for cancer is exactly as you say, because you're inhibiting migration.
Right.
Yeah. And we didn't know about it until this paper. So this could be, in fact, exactly as you're saying, the reason why these antibodies aren't really useful in cancer treatments.
But the other antibodies are okay.
Yes, but the other antibodies
in a
are
subset of patients.
In a subset,
right. Yeah.
I wonder why. Maybe there's something to do with migration there too.
Well, that one could be more specifically impacting the T cell function.
Yeah. So the one thing we didn't mention was that this is only one pair of many pairs of receptor ligands that dampen responses. So cancers can upregulate a number of these ligands that are, the receptors are expressed on T cells, and they can impact and impair T cell function. Plus they can donate their mitochondria if they so chose to. But yeah, there's lots of ways in which cancer cells are dampening T cell responses. So it could be simply that cancer has not upregulated PD-L1, so it isn't working by that mechanism. So if you try and use the anti-PD-1 to rejuvenate the T cells, they weren't impacted in that way to begin with.
And they use their antibody blockade in the mouse. And they use either an antibody that blocks PD-L1 or an antibody that blocks PD-1. So the antibody that blocks PD-L1 is really going to be blocking that cis interaction because of where it binds. So PD-L1 cannot interact with things like CD80 on the same cell, but it still could interact with PD-1 on a separate cell. Or they use an antibody that blocks PD-1. So now you're blocking the trans interaction, but potentially not the cis. And if they block PD-L1 with that antibody, they don't see dendritic cell migration. If they block PD-1, there's no change in dendritic cell migration. So this, again, Vincent, could be sort of why there's a difference in the outcome of patients with some of these antibodies. Then they get into some of the same questions that Cindy was asking. Where they start to think about some of the questions about cis and trans. And they also spend some time here thinking about, are these antibodies on the cell surface somehow blocking access of chemokines to chemokine receptors, and that's why all of this migration is blocked. And so they look at, they go back into an in vitro assay and look at cells moving through those little pores across a well, through a trans well, in response to a chemokine. And they see that they can get migration in response to chemokine if they use an antibody against PD-1, but that goes away with their anti-PD-L1 antibody. And it looks pretty similar to the knockout cells with the genetic deficiency.
Are you going to talk about these? So they did one control experiment here that I thought was really cute. And that was they tested in the presence of anti-CD45 antibody.
I wasn't specifically going to talk about that one, but go for it.
Oh, yeah. So if you think about it, what they were trying to ask is, could just adding all this antibody bind up the surface and sterically hinder the access to the chemokine. And so they used anti-CD45. So CD45 is expressed at high levels on most immune cells or all immune cells. And even at a high density, if you add antibodies to that, it still didn't block access of the chemokine to the chemokine receptor. So it was something specific about the PD-L1 versus just any generic steric hindrance of access to the chemokine receptor. I thought that was a nice control.
I thought that was, yes, I definitely thought that was a nice control. In general, I was very impressed by how thorough they were with a lot of controls for a lot of different things, even, that I never would have thought about.
Yeah.
I was pretty impressed with that thoroughness.
The first author must have had good training.
So they now wanted to get at some of those questions about knowing whether this is cis or whether this is trans. And so if this is a cis interaction, we're imagining PD-L1 on one dendritic cell interacting with CD80 sort of adjacent to it on that same cell. And so first they look at migration of cells that just have PD-L1 and are missing CD80. And there's no migration. Not a surprise. But then they actually mix together cells that are CD80 deficient and PD-L1 deficient. So you wouldn't be able to have CD80 and PD-L1 interacting on the same cell here, but you could still have trans interactions between the two different cell types in this experiment. And they don't see migration in this case. So if they don't have CD80 and PD-L1 on the same cell, the cells can't migrate. They also use cells that have PD-L1 and they give them soluble CD80 protein. So here, you have both extracellular domains present, but you don't have the transmembrane domain and intracellular domain present. So you can get the protein-protein interaction, but not the intracellular signaling. So that kind of gets at that question that Cindy was asking of, do you need the intracellular parts of the proteins together, or is just extracellular binding enough? And again, they don't see migration here when they use soluble CD80 or when they use soluble PD-1. And so it really looks like you need the transmembrane domain and the cytoplasmic domains of these proteins for this interaction. They do some additional work with some mutations, and they sort of show that they get some ideas of residues that are involved in this interaction, but they don't fully come up with exactly which residues on both proteins are involved. But certainly they can say that you need the CD80 and the PD-L1 on the same cell to interact and get intracellular signaling to that dendritic cell for migration. So now they go to biochemistry. And they, here, like I said, they're basically mapping the surface of PD-L1 and mapping the surface of CD80 and then making mutations to try to figure out, is the intracellular domain important? Is the extracellular domain important? Which particular residues are important? They see that the extracellular domain residues that allow for the two to interact in cis are really critical. It's a little bit unclear what interactions are necessary in the intracellular domain. And I think this is
But they are necessary though, right? There's some interaction, could be bridging with some adapter molecule or who knows what.
There's some intracellular interaction that's important, but exactly which residues are critical is not completely clear here. So now they go into a mouse tumor model. And specifically, they're looking at melanoma. So again, we're sticking with skin throughout this paper as our tissue. And they they have mice who have melanoma. They treat them with anti-PD-L1. And they see that there's not good dendritic cell migration into the draining lymph nodes when they treat with this anti-PD-L1 antibody. And that is similar to, and so really there's no sort of enhancement of anti-tumor activity. The mice still have the tumors, the mice still have cancer. This looks similar to kind of what you would see if you use this same antibody in cancer patients, and that it doesn't do anything about their cancer.
So it's used in melanoma?
It is used in a few different cancer types. I don't know exactly all of the types that anti-PD-1 is used for, but it's used in quite a few. I have strenuously avoided saying the compound names for any of these antibodies because I can't pronounce them.
I wondered about that. I was trying to pronounce it in my head.
Yeah. So the anti-PD-1, which a lot of people use, is pembrolizumab.
Mm-hmm. Yes.
So people may have heard of Pembro. I know Keytruda or Opdivo are the ones. So those are the ones that work. The ones that interact on the other side that don't seem to work as well are atolizumab, along with two others.
So it is melanoma, non-small cell lung cancer, bladder cancer, kidney cancer, head and neck, squamous cell carcinoma, Hodgkin's, colorectal, and gastric. Lots.
I knew it was a long list.
Wow. Merkel cell carcinoma and hepatocellular carcinoma.
It has been very useful in the cancer immunology field. And so why the other side of that interaction doesn't have an effect? Well, if you block PD-1, it has an effect in blocking PD-L1, which was thought to be its main ligand, doesn't work, was sort of the question here. But the final thought that they had was, okay, so we see that if we block dendritic cell migration using our anti-PD-L1 antibody, this new PD-L1 function we've seen, we're blocking dendritic cell migration. That's not so good in cancer therapy.
Yeah.
And as Vincent points out, you wouldn't want to block dendritic cell migration in cancer therapy. And so it's not surprising that this isn't working. But we could imagine situations where we would want to block dendritic cell migration. Not in a cancer situation, but say in an autoimmune disease or in an autoinflammatory situation, we in fact might want to block that self-antigen from getting transported to the draining lymph node. So if we now take this drug and use it in a mouse model not of a tumor, but of an inflammatory disease, will this drug actually be useful? Will it still block dendritic cell migration, and will it have efficacy in this mouse model? And so now they switch to a psoriasis model where they treat mice with a toll-like receptor drug that induces signaling through toll-like receptors, imiquimod, and potentially induces skin antigen to go to the draining lymph node. And when they use this psoriasis model and block PD-L1, either with their antibody or with their mutation, they ameliorate the psoriasis in the mice. So they basically have stopped the psoriasis in the mice, and they show that they are not getting dendritic cell migration to the lymph node. And they show that when they have fewer dendritic cells in the lymph node, they have fewer Th17 cells being generated in these mice. And so while this is giving an idea that, okay, now we might have an understanding of why this antibody was not particularly useful in a cancer setting, even though with our previous knowledge of immunology, it should have been, we can see another situation where clinically this antibody might in fact be useful because we want the dendritic cell migration block to happen.
Yeah, I liked that. I thought that was fun. That was really clever.
Yeah, I liked that it sort of started with clinical observation, then went into mouse models, went all the way to biochemistry, and then went back to a clinical use.
Yes.
So when they use CAR T-cell therapy, right, to direct T-cells to tumor antigens, how do they get over the PD-L1 issue?
They have problems sometimes. They get shut down. Like you get you deliver these T cells and then they don't work that well.
So sometimes they do combined therapy, CAR and checkpoint?
And that's why the CAR T cells really are effective with blood-based blood cancers.
Correct.
Because you don't have to deal with this, the tumor environment.
That's right.
Mm-hmm.
Yeah.
Yeah. And that's also in some ways why some of the CAR NK cells or CAR other cells might be useful, because they don't have PD-1 on their surface. And so they wouldn't be as subject to the exhaustion mechanisms.
Oh, and the blood cancers, they probably are not innervated, right?
Right.
So
True.
maybe they're not swapping mitochondria either.
Right? Maybe because of the flow of blood, you still have
it would
some
be
connections.
really hard.
Less connections.
That would be
right?
really hard.
Very long nanotubules just spindling into the void.
I imagine this like you're sitting in a float, right? You're floating down one of those lazy rivers or one of those water flumes, and you're trying to hold a straw in your mouth, and the other person's trying to hold it in their mouth at the same time.
You're not going to steal the boba from their boba tea.
You're not going to steal a boba.
Well, you could adhere to the endothelium, with the right receptors.
Yeah.
But the innervation is going to be the result. Very interesting. Thank you, Brianne.
Yeah, Brianne, that was great.
All right, that's number 95. Immune. Coming up on 100, folks.
I know.
We gotta think about what we're gonna do.
If our numbers are right, it's December.
Do something.
Yeah.
December, right.
Ooh.
It's the end of the year. I have to think about that.
Yep.
No, you could always come here and
we should
do
come. Actually, that is
here.
we should come to New York.
I can come there.
We could have, you could easily.
Get a train.
We could have a little audience here at the incubator.
Oh, that would be so fun.
That would be fun.
Because we could probably fit 20. We've been talking about doing some of those here as fundraisers, but we could do them. For 100, that would be fun.
Let's do it.
And I bet local people would come. So let's talk about it. Yeah,
I think they would.
Number 95, questions and comments, immune@microbe.tv. If you enjoy these programs, we'd love your support. Microbe.tv/contribute. Cindy Leifer's at Cornell University. Cindy Leifer on Blue Sky. Thank you, Cindy.
Thank you.
Steph Langle, Case Western Reserve University. Stephanie Langle on Blue Sky. Thank you, Steph.
Thank you. This is great. Thanks for the papers all.
And Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Everybody's on Blue Sky now.
Yeah.
Even Microbe TV, microbetv.bsky.social.
Awesome.
How about that? I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.